So tonight I am delighted to introduce to you Jane Greatorex, who is not only Lucy Cavendish College's own senior tutor, undergraduate and graduate tutor, and director of studies in preclinical medical and veterinary sciences, but also has a long career in academic and clinical sciences, specializing especially in blood-borne viruses. Until 2017, Jane was responsible for streamlining and improving HIV diagnostic services at Addenbrookes Hospital. And she was also, in 2015, team leader in Sierra Leone during the Ebola outbreak. And she further remains involved in a number of research projects, partly involving HIV and also influenza, for example. Most recently, of course, everyone has been asking her about this COVID-19 related stuff. And so she's been not only very busy as a senior tutor, helping to make the college or to help the college deal with COVID-19, but also she's been on the radio and interviews, etc., etc. And this is also going to be the topic of her talk tonight, which is entitled, and that's a bit of an update from what was on the website, Viruses and Pandemics, Lessons Learned. So without any further ado, I'll hand over now to Jane. Welcome, Jane.
Thank you, Henriette. And I'm actually going to start with an apology because I've changed the title of the talk again somewhat. I've been thinking about this for a while because as Henriette says, I have been asked a lot of questions about coronavirus. I've been asked for sort of common sense answers to what we should be doing. Should we be physically distancing? Should we be wearing masks? Should I send my child to school? All these sorts of questions. And I'm very, very happy to answer questions around that at the end. And I'm sure, people will have questions. But actually, what I thought we might do is just take a step back and remember what got us here in the first place, and that's actually the virus. So I'm going to concentrate on the virus and go through its life cycle and what it does, how it spreads, to try and help everybody here understand why we take the mitigations we do and why I say, when I'm asked those questions, the responses I give and other experts give. And then also, this is a talk like no other talk I've ever given. I mean, ordinarily as an academic, you are talking to people in your own field, usually quite narrow fields or often quite narrow fields, or in the college context, I might be talking about my life or travels or something like that, usually to other academics who are simply interested, which is great. But this time we're all involved, and actually everybody you speak to is affected personally by this in some way or other. And what this virus has done in spreading around the world has made a situation I don't think that's ever been the same, not in the sheer reach of this virus globally. So I'm wary of that. I'm aware of that as I go into this talk, and I will try and answer all the questions that you have for me, I hope, at the end. But I'll do some slides, and then I may or may not sort of pop back in between, but we'll use the slides just to illustrate my talk. So yeah, up close and personal, because I want to talk about the virus. I am going to jump to two slides that just talk about the state of play and why we are where we are, or why you're sat here listening, really. This is this morning's data from the European Centre for Disease Prevention and Control, and it's taking us right up to the sort of present day, of course, this morning, not 10 a.m. And we're the green bars, so we're ahead of everyone else, as we're probably all aware. And this is this morning's data as well for Europe. And we lead on cases, we lead on deaths. If you take population into account, then things look slightly better. There are other countries less populous than us that end up having more cases per 100,000 of population. Deaths we still don't do very well on. And there are multiple reasons for that, and that's not the point of this talk, but the point of these two slides are to remind us of just why we need to be concerned, and why we have to continue to take the mitigations that we have taken and may yet need to return to. So here it is. I'm going to show you 4 viruses actually on this particular slide, but this rather lovely, I think, 3D structure is from the Center for Disease Control in Atlanta, and they kindly give out these slides for media. This is lovely in that, well, I think it's lovely, in that it has on it all the proteins and shows very nicely and clearly the structure of the virus. Now, there are other RNA viruses that cause pandemics, and these include this one, and there'll be a sharp intake of breath from the virologists in the audience at this moment in time, because I've chosen a slightly controversial picture here. That virus doesn't look, I guess, to some eyes, much like a virus, because it's a kind of funny shape. But it's a filamentous flu virus particle. And influenza has caused 3 pandemics in the last 100 years, the most severe of which, as you all know well, was the 1918 pandemic, when, you know, 1/3 of the entire population of the world was infected with the virus. And 500 million people died as a result, which is unimaginable. There we go. HIV, my favorite. Sadly, a pandemic that's ongoing. And we forget this, because here in the UK, we manage this virus. We manage it by treating patients that are diagnosed with it. And they live long and almost normal lives. They die of things that we all die of, mostly. They will be susceptible to COVID. And then last but not least, I had to put Ebola on here. It has not caused a pandemic as yet, but it served as a very important reminder to us. It caused a large outbreak, which affected mostly 3 countries in Western Africa, but people from those countries traveled and became ill in other countries, and there was onward transmission. And that is a reminder that air travel is a major driver. We don't need a reminder now. We can, we know what happened. From the outbreak in Wuhan to crossing the world in a matter of months. If you think back to swine flu, I'm always mindful of the fact that I follow a program called ProMed, which a lot of the scientists amongst you will follow, and that's around the world reporting of outbreaks as they happen. And as the outbreak happened in Mexico, 2 or 3 days later, we had a couple who'd honeymooned in Mexico, flew to Scotland and tested positive. And that was the virus in the UK, just like that. So we know how fast these things travel. One other thing about these 4 viruses that all of you will be aware of is that they have RNA genomes. And we'll come back to that RNA genome. But what does that mean? Why are these 4 viruses associated with either large outbreaks or pandemics? And it's their ability to mutate, to adapt, to change their host. So we know that influenza has got a segmented genome, and it mixes and matches its genomes with avian influenza viruses, swine influenza viruses, and the human virus. And therefore it can adapt quite easily it can adapt to living in different hosts. HIV, we now know from doing genetic studies and going back, was a zoonosis also. So it transmitted from monkey or chimpanzee and gorilla species into the human species, into Homo sapiens. And we are reasonably sure that SARS coronavirus 2 came from bats, as we are reasonably sure did Ebola. So all these viruses able to mutate, able to adapt to different hosts. In the case of HIV, if you think about that adaptability, think of how difficult it has been to make treatments to that virus. And that's its ability to, as fast as we find a new treatment, mutate and adapt to that treatment. And they do this with our own immune systems too. And close and personal with this virus. So here we go. This is the outside. And these, when they talk about the coronavirus, it's this crown of spike proteins that have this sort of funny structure. I think it looks more like butcher's hats, the old-fashioned white ones. But anyway, crowns. And then there's membrane proteins, these little orange guys, and then fewer amounts of these envelope proteins stuck into the membrane of this virus particle. I am going to show you one. Oh, don't flinch at this. It's there for the virologist, but it's also there to remind me and to amaze you all, I hope. That is a tiny old genome, and yet that virus can get into the cells in your body, replicate, produce thousands and thousands of new virus particles, and get out and find a new host. That's about 30 kilobases. Your own genome is about 3.3 gigabases. So it can replicate itself with a very teeny genome. And that genome packs an awful lot of punch because it produces the, fine, I don't have a pointer. It produces the proteins that will cleave the initial proteins. I'll show you in a moment when it goes into a cell. It produces the proteins it needs to replicate, and it produces some of the structural proteins that are going to do the binding to the cell it's going to infect. The rest of the time, viruses use you. They use the mechanisms of your own cell to replicate themselves. They pack the minimum amount they can or need to, and use you to do the rest of the business. So we'll just take that structure. This is a nice cartoon. Most of these come from an online book, which we hopefully, when we come to the discussion, I might talk about a little bit. So this is looking inside the virus particle here, and you can see so there's the spike proteins on the outside, these big turquoise ones. The envelope is, the membrane rather, is the purple one, and then there's these fewer numbers of envelope proteins stuck into the viral membrane. And inside is a single strand of RNA, and that's its genome, that genome that you've just seen the map of. And it's coated with a nucleoprotein. And if we just take a closer look at the spike protein here, you don't need to admire it too much, but you can see the sort of crown-like thing. And in there's a groove in this protein. And you just think that we didn't know the structure of this protein 6 months ago. That's how fast the science has developed around this virus. But we do know. And there's a receptor, sorry, there is a protein binding site here that binds this molecule. And this molecule is where all the trouble lies. So this is the angiotensin converting enzyme 2. And it is present on a lot of cells, but chiefly you'll recognize these: the lung, the kidney, the liver, the heart. It's good, isn't it? All those organs where you really don't want this virus to be replicating and binding and attracting attention. And we'll come to that in a moment. So angiotensin 2. So the reason we have, why do we have these receptors over loads of cells? What that enzyme is responsible for on the surface of your cells is it modulates or catalyzes one form of angiotensin into another, and it takes it from vasoconstriction to vasodilation. So it's, in a way, it's a good thing, but it's a virus binding site as well. So when the virus binds, so it binds these receptors that are found on a lot of different cells, but importantly, those cells I've already mentioned. And the virus particle is taken into the cell, and then the membrane, is disrupted. The virus particle gets into the cell, and the genome, that single strand of RNA, is then used as a template. So RNA is a template to produce the proteins that then go on to do the replication. So those proteins will then replicate the viral genome and do two things. You translate more proteins from your RNA, and what you get is lots of viral genome being produced, but also all the structural proteins of the virus. And in order to do this, it's using the nucleosides within your own cell. It's using a lot of enzymes and things within your own cell. It's basically parasite. And then what it does is it takes its genome and it takes that gene, the RNA up, it mixes it up, it binds it around with sorry, trigger happy. It binds it around with the structural proteins. The membrane forms around it from within your own cell. And then it gets pinched back out and produces lots and lots, thousands and thousands of virus particles, and goes on to infect other cells. So here, what I want to just briefly talk about is what do we see and why do we see it? So bear in mind that if you think about an ordinary respiratory virus infection, there tend to be upper respiratory tract, you have a cough, you feel rubbish for a few days, and we'll come to that in a moment. And then you get over it, don't you? But the problem here is that that virus can bind to cells throughout the body, and it can get down and into the digestive tract, it can get into the heart, the kidney, the liver, and so it's going to cause far more far-reaching effects. It can also get into the central nervous system. So what you see, first of all, because bear in mind that virus is going to bind in the respiratory tract, first of all, are the symptoms that we're familiar with: fever, headache. Hemoptysis is basically, blood, coughed up blood. You get a cough, again, pain. This is all sort of normal viral type symptoms. And for some people, that's the end of it, that they get over that. I think a lot of people report feeling pretty ill for a while, but they will get over it. Sadly, the older you are, the more likely you are to develop a more severe infection. And you go on and get the shortness of breath that's also associated with this. I think there's the lack of taste and sense of smell. That's an earlier symptom. But you can go on and you can develop pneumonia, septic shock, renal failure, and diarrhea, because remember, it was binding to intestinal tract cells as well. So why is this? And this, if I'm honest, is where there's an awful lot of research still ongoing. And I just want to I'm going to pause here just for a moment, if I can, and talk about this a little bit, because this is something I get asked all the time. What's the current state of play? Why does it do this? Why does it do that? And if there's two things here, we're 6 months in, possibly 7 if you think that really the Chinese investigators started talking and thinking about this and researching about this earlier in December. But it's a massive amount of work that's going on globally to really pull this together, to really, and you can't just, there are a lot of cases, there's an awful lot of data, but as fast as that data is generated, it has to be processed, it has to be assessed. Not all the data, and when you think about data, the scientists, the social scientists amongst this audience, you know that there's one thing designing a study and thinking of how to control it and how to do everything the same way, how many controls you're going to have, how many patients you're going to have, what age groups, what are the comparisons, what are the normals. That doesn't exist when you're trying to work in real time in the context of a pandemic. So it's amazing that within weeks of those first cases in Wuhan, the Chinese had shared with Australian scientists the genomes of the first isolates that they had, that we had a sequence immediately, and people could start thinking about that. What does it mean? What are the proteins? What does this virus look like? Where does it come from? And even now, I'm asked a lot, so how do you compare, how are we doing testing-wise? Well, you can't answer these questions because we're all doing things differently, and we've developed tests differently, we assess data differently, we report data differently, as those of you that follow the news will know well. So it's hard to answer these questions, and to be honest, if you think back to, I think swine flu is the one that I worked on the most of the pandemic viruses in a clinical setting. It probably took us 10 years before we had a really good understanding of the epidemiology of that, of we still work with that virus. We still wonder about that virus. What would it take to make it more or less virulent? Why was it not so virulent? In the context of the pandemic at the time, I mean, I was employed at the time on a grant funded by the NIHR, and we were funded to look at transmission. How was that going to happen? And we were, you know, it was over and done with by the time we'd actually got the data out properly and published and peer reviewed. It was probably a year after the actual end of the main pandemic, and there was a second wave then, I remember that. So what has changed? And I think something that's changed now reflects back on some of those images I've been showing you, some of the diagrams I've been showing you. There's much more preprint publishing, and that's, I think it's extremely exciting. It's both the bane of my life and it's the boon of my life. You know what's going on because people are putting data up there all the time. So when you read in the paper, newspapers are naughty. They don't say where that data comes from. They say things like, oh, a new treatment, look at this, fantastic. And the paper has not yet been, it's in pre-print. It's not been peer reviewed. It is literally not rough draft. It's ready, not always. It's ready to go to a journal for assessment. And I speak of having done this myself, but it has not yet been through normal review. Now that those diagrams are different, there are some press that actually have just put up much more rapid sharing of both review articles, book articles, diagrams, you name it, resources. But they've all been reviewed, and that particular one has, so I'm confident I'm showing you the real thing. But others, you know, studies that have been published, have then not actually made it to real press, to actually go out in a journal. So you need to be careful. That said, what it means is it drives collaboration between scientists. And if you see something and you think, goodness, that's what we're working on, or how fascinating, you can get straight on it, get in touch. Actually, we have the cell line that you need. So I'll get back off my pulpit, but just to remember, when you are reading on the BBC in particular, the BBC, I won't name them, most of the big newspapers, they are talking about preprint data. Right, let's get back to my slides, sorry. Okay, back to our little human. So back to the human, and then back to, really, I hope. Yeah, back to this slide. This is actually from a preprint book, but it's quite nice and it's quite simple. If you look at the pathogenesis of SARS coronavirus 2, there are hundreds and hundreds of papers already, and they'll be talking about in the liver, in the heart. There's lots of disagreement. This is basically what happens, and then there are many layers of complication on top of this, and you know, the individual patient alone. But if we start at the top, there's our little virus. It's a rather funky looking version here, but that virus gets into the cells where we don't want it to get. So the lung infection, where you might expect it to get to from the upper respiratory tract, but beyond that, via the blood, we find it in the blood, to the intestine, to those other organs, where it binds. And what it does there is it triggers so it's binding to that ACE2 enzyme. And what it does is it triggers, the cells trigger a signal. They go, goodness, we're infected. And so they release these chemicals, cytokines and chemokines. And what these are the messages to the immune system to start activating, to do something about this. And specifically, they activate what are known as T cells. And the T cells are designed, or certain subsets of the T cells are designed to respond to virus infection. And they do that, and then guess what they do? They produce more cytokines. And so it's a you know, cumulative effect of more and more cytokines being produced. And unfortunately, that's the pathology. It's those cytokines that cause all the disease. They may stop the disease, as it says here, but they may go on and keep producing more and more exudate in those as part of that storm. But ultimately, as they attack the tissues, they can lead to tissue failure and organ failure and secondary infections as your immune system attacks itself. So that's where we think the problems lie. So this is a tiny virus, 30 nanometers. That 30 nanometers, let me put that, to put that into perspective, you could get 3,000 of those guys into one. If you just pull a little strand of hair from your head, the diameter of that hair, you've got 3,000 virus particles across it. So these are tiny, tiny things, and yet they cause this terrible effect. If they get on, if they get past your immune system. So just to talk about, I have to mention transmissions. How do we stop? What causes the transmission? Obviously the cough, the sneezes, the giving out of the virus. And then how do we transfer it amongst ourselves? We know this, you all know this. I hope everybody has been so well and truly educated, but close contact, surfaces or fomites, and airborne transmission. Now, you might think, well, respiratory virus, that's the most obvious. Surely it isn't. The close contact is where the most virus is. That's the large droplets that you cough or you speak, some people, ordinarily. And then the airborne transmission is the much, much tinier particles that come out when you breathe. And there's a whole host of media, there's review articles about airborne transmission and its role. We know it occurs. It's been found, I'm sure there's at least one person listening that knows about the quires and the transmission of viruses. And we believe that was an example of airborne transmission. Surfaces and fomites, we know, you find the virus, you can isolate live virus. So the mitigations, and I'm putting this in at the end because I hope that you can see why we need to stop it, and I hope that you can see how we can stop it. So you have to, and this still holds. Hand washing is the one mitigation that holds up around the world, everywhere. You are simply, if you yourself have it and you put your hand up to your mouth and then stick your dirty hand on the surface, then you're going to leave that live virus there. Because you've put your hand close to the face, you're getting the large droplets, you're getting more of those viruses in those droplets. Not my dog. Then the 1 meter rule. This is me being naughty because we currently have a 2 meter rule, don't we? But physical distancing is important. I do believe that if we're wearing masks, unlike those people in that picture, that's a UNICEF, that UNICEF advocate 1 meter distancing. I believe that there's plenty of data now, especially in recent weeks, that says that the wearing of a mask and a 1 meter physical distancing would work just as well as 2 meter distancing. So that's my little message for the day, University of Cambridge. And then lastly, the wearing of masks. I think this is something that we are going to just have to get used to. Everyone else does it. I do not understand why the British find this so difficult. If that is the means by which you can sit and talk to somebody in a room, then what's not to like? Go into shops, be in crowded places without transmitting, without passing on that virus. If we don't, if we start to relax things too much, there will be a second wave of this virus in the autumn. It might be somewhere like Cambridge, which has a relatively low R0, which I hope we're all familiar with now. That's the ability to pass the virus on to more than one person. Then we might be all right here, but we won't be all right once we're in the country. So I think I'm going to end. I suspect there's quite a lot of questions. This is a quote from The Plague, which I thought was appropriate. But and the reason that it's appropriate is because of my belief that we will continue to learn from this. I hope that this is unprecedented working together of scientists at this moment in time. And I believe that there's such a will to sort this out that we will be able to certainly mitigate against things, hopefully get a vaccine eventually, and eradicate it, or at least drive it down to the levels of other respiratory winter viruses. Thank you.
Okay, thank you, Jane, for giving us all these, what you call lovely pictures of viruses, where other people might maybe say scary, but fair enough. What I'm going to do is I'm going to pick up on all the questions that have already come in, and then I'll ask you to give an answer, and then if those that have submitted the questions have follow-on questions, please feel free to take the floor. So the first question that's come in tonight, let me just make sure that that's indeed the first one. Yes, I think it's from Suzanne Doyle-Morris, who says, I'm aware that there are plenty of viruses that we still don't have vaccines for, even after many decades of research. Are we more likely to find a vaccine because of the sheer number of researchers working on this? Or what are other variables that will increase or decrease the likelihood of us finding an effective vaccine?
That's a good question, Suzanne, thank you. There are many viruses out there. Oftentimes there isn't the driver to produce that vaccine. You know, Ebola was a case in point, wasn't it? That it affected so few people, even though it's a terrible virus with a sort of more than 50% mortality rate. But there wasn't the drive until all of a sudden there was a possibility that it might go around the world. And then there was a drive, and then bingo, we've now got some very good vaccines actually against Ebola. For this particular virus, I think, yes, Suzanne, you're absolutely right. There's a massive amount of work, and I would say the big positive is that there's so many people working on it. There's so much collaboration. The studies are being, you know, there's early studies, but then there'll be much bigger drivers from, you know, the commercial companies and collaborations between them and academia. That I would hope that we will get there to something that will produce immunity. I say it cautiously because we've been working on vaccines to the first SARS virus, the first SARS coronavirus, which people will remember came from, was it civet cats, that one? And then the MERS coronavirus, which, you know, it's still a major source of concern for us. It still causes cases every year. We still worry about it when we get people returning from the Middle East. That came from camels. These have proved very difficult to produce vaccines to with good lasting immunity, and that's sort of, I think people will have heard echoes of that maybe in the press. That said, let's hope that, you know, time and not time, but the sheer number of researchers is on our side this time around.
Okay, Suzanne, are you happy with that answer? Or did you have any follow-on question? I'm just going to assume if I don't hear from you that means you're happy with it. Okay, okay. I'll move on to a next question then, Jane, from Victoria Harvey, who picked up on your remark on real-time studies and is asking when or if the government at some point is going to relax shielding categories. Is that likely because of emerging data or better care management, or is it actually way too soon to expect that type of data to be available to make those decisions?
It's a good question. I mean, I don't know the absolute answer, Victoria, from, you know, our government currently. I would imagine, however, that it will be based on data across, you know, there's so much data across the world globally on shielded people and their risk factors. I mean, we know already that they're constantly I think, not redesigning, redesignating, that's it, redesignating some of those vulnerable categories and removing them from the list. The very shielded people, for the time being, will remain so. Yes, there are, I mean, as case numbers go down, that's the other thing. You know, if we can drive that down, then the risks become much less, don't they? I think there was a fear that people might remain shielded until vaccines were produced, but one would hope that if we can get that R number right down, then that won't be the case.
Thanks, Jane. I think that was my question. It's how are they nuancing it? Because I noticed they've taken off a lot of, you know, the more obvious ones like respiratory problems seem to have come off the shielding list, and less obvious ones are still on there. So I'm wondering if that's a result of the data studies they're doing on the current patients, or if that's...
Yeah, absolutely. Sorry, Victoria. Yes, it is. I didn't answer your question properly. It is, and it's also based on data from other countries that are, you know, a little bit ahead of us, and they've had more cases, you know, up until recently. So yeah, it's a mix of the two. But I mean, as fast as they can, they will, you know, where they see that people could be out and about, they will say so.
Brilliant. Thank you.
Okay, so the next question that was submitted, so to speak, was from Lucy van der Wiel. And her question is, could you discuss the potential effects of the virus on the cardiovascular system? More specifically, could a viral infection increase the likelihood of bleeds from malignant wounds?
Oof, I don't know about the malignant wounds thing. I mean, we know that cardiovascular disease is a risk factor for COVID. So some of the injury that you already see in cardiovascular illness does not then help the body respond well to the actual viral infection. So cardiovascular disease in particular is a major risk factor. And that sort of muddies the water when you're talking about things like type 2 diabetes, where people have got, you know, a higher likelihood of having that disease. That's not answering your question terribly well. I'm not a cardiologist, so I can't speak specifically to bleeding. Sorry.
Okay, Lucy, are you happy with that response?
She says, thank you very much. I can see that.
Okay, good. Okay, good. Okay, so the next question is from Leticia. And she says, considering people of the same age, do we have any idea why the severity of COVID-19 is so variable among individuals? I've heard men are more likely to have more severe symptoms. Do we know why? Is there any possible link with previous exposure to viruses of the coronavirus family, or?
We're looking at all sorts of things. I mean, there's some massive studies going on. Actually, here in Cambridge, there's a massive study that's taking all the data from everything. And I mean, when I say all the data, I mean all the hospital data on the individual patients, everything you can think of, blood groups, all their chemistries, all their hematologies. And then just searching for profiles to find out what, you know, how did they get there? Why? What is it that makes them a risk factor? And can they then direct treatment and so on? Those sorts of studies are being done nationally as well, looking at data from all the patients. It's hard, Letitia. I mean, even with the sheer number of cases, what you really want to do with your sort of question is say, right, here's, you know, 2 women, they're the same age, they're both healthy, got the same blood group, you know, why did one get the virus and the other and respond badly to it, and why didn't the other? Some of these questions that are so difficult, and like I was trying to allude to with the type 2 diabetes, it's complex, you know, that you'll have multiple risk factors. The clear ones, the ones that you keep hearing that come through from every study, are age, absolutely. And diabetes sadly still is. I think 1/3 of all the people that have died type 2 diabetics, something like that. It's not good risk factor. But first and foremost, I mean, what for instance, the Diabetes UK pushback is that age is the biggest risk factor. So it's something, I think that's more likely to be around your immune system. So you asked Letitia, didn't you, what if people were the same age, then there are other more subtle factors there. And it's hard to say. I mean, I've listened to people saying, well, so-and-so seem to be healthy, but you don't know that. Very difficult, isn't it?
Okay, Letitia, are you happy with the answer?
Yes, thank you very much. Yeah, I mean, I know it's a big question and we don't know, but I keep wondering. Thank you.
Yeah.
Okay, great. A question from Jane Robinson. Does COVID-19 affect the brain? I mean, I would answer that and say it affects all our brains because our brains have been filled with this thing for the last 3 months, but I'm sure you have a more scientific answer, Jane. So
there's evidence of neurological disease as well. Yes, I don't know enough about it, but there's certainly studies where it flags up. Yeah, I mean, the sheer numbers involved here mean that there is so much evidence and so much data that some things like, I guess one of the examples would be the that syndrome, I've forgotten the name of it, in children, you know, but it's so few cases, they're found because there's so few cases with that syndrome, but they're found because there are so many cases. So my answer is partly that this data will bring all these things up. We just can't answer all the questions right here, right now. If we could, we would, basically.
okay, thanks, Jane. Jane, are you happy with that answer? I'm going to assume that it's a yes if I don't hear from you. Okay, then I'll move on to Barbara M. King, who says, I've been reading about the healing effects of breathing techniques and cold temperatures to combat certain viruses. How do the scientists see this? And is there an appetite to explore this as part of research?
I don't know is the answer to that one. I mean, with
mentioning field studies with E. coli.
Yeah. I mean, I can imagine that things like breathing technique would help ease. I think a lot of, I mean, the straight answer is I don't know of that being used in any research studies. I can imagine that breathing techniques are used to help people manage, you know, the shortness of breath when it goes on for a certain amount of time. It must be fairly distressing, particularly in the elderly. But I don't know about it as a treatment, no.
Okay. Okay, Barbara, did you have a follow-on question from that, or?
No, thank you, Henrietta. I think it's a tough one, Jane, but I ask it because it popped up on something this morning, and there is I think he's a Dutchman known as the Iceman, who's claiming that, you know, this, if you follow his techniques, essentially, we wouldn't have such an outbreak of COVID, and that actually it ought to be considered more considerably by the scientists. So I thought I'd ask you the question.
No, fair point, but it's not a sort of, I wouldn't care to try it, though, you know, just like that. In the case of, in the context of COVID, new treatments tend to be done on severely ill patients, basically, because you've got to try something.
Although I think that the comment on the temperatures was quite interesting. I mean, I'll send you the link anyway, just for you to have a look at.
Thank you.
Thank you.
Okay, thanks. Let's move on then. There's a question from Nadia, who is asking if there is any evidence of mother-to-fetus transmission during pregnancy.
So there have been babies born that appear to have COVID. I don't know that they've absolutely ruled out that it didn't happen during the actual delivery. So, that's a good question. I don't think anywhere I have read of definite vertical transmission, but certainly babies have been born where mothers have tested negative. That's a whole other ballpark though, because tests, don't get me started, you know, on deciding whether testing's working well or not.
Nadia, do you want to have a follow-on question on that, or you're happy?
It's great. Thank you so much. I'm good.
Okay, thank you. Right. Let's see. The next question comes from Christine. And she said, given what you said about air travel as a major driver in transmission and the success of other countries who have closed entry or insisted on quarantine, do you think the UK should be restricting entry at all airports?
I think there's other things we could do. I think we could be temperature testing. I think